Welcome to Education Day of Timmy's annual Global Health Week. Today we are proud to speak with Dr. Ngo, an assistant professor in the Department of Biomedical Engineering who earned his doctoral degree in biochemistry and molecular biophysics at the California Institute of Technology. Work in the Ngo lab applies the principles of evolution, chemistry, and engineering to develop new tools for measuring, visualizing, and controlling biomolecules in living cells and organisms. We're going to go ahead and start the questions off. Our first question is, how do viruses enter and use our body?
Into the body. So I guess it depends on the type of virus through different routes. The reason why this particular one is especially transmissible is because it's a respiratory virus, which means the primary site of infection is the respiratory system, the throat and the lungs, and it is transmissible therefore because droplets that are generated in the lungs and comes up when people cough and can be a route of transmission. This is in contrast to, for instance, HIV, which has a different route of transmission because its primary site of infection is T cells. And so that generally is a blood-borne virus that is transmitted through the blood or other bodily fluids, but not so much through respiratory particles, etc.
How does the virus harm the body?
Okay, so the virus in this case uses cells that express a certain receptor called the ACE2 receptor, which stands for angiotensin converting enzyme. And it uses that protein that's on the surface of airway epithelial cells to enter into the cell. And then once it's in the cell, it replicates many, many copies of itself. And then it also shuts down the host defense system so that once the virus enters into the cell, it produces new proteins that will replicate its genome and form new viral particles so that it can go on and infect other cells. But in addition to that, the virus also encodes proteins to shut down the host cell's gene expression program such that it's not able to signal to other cells to inform them, alert them to the presence of viral infection. What ends up happening is that those cells, there are several hypotheses, but those cells end up killing that cell, and then there's a subsequent immune response that is an overreaction, and that's what patients typically suffer from. So the symptoms of the viral infection, in this case, is overreaction in the immune system, or in most severe cases, death to the overimmune reaction.
But for a respiratory virus, I think most people understand that it's transmitted via inhalation of aerosols or droplets entering the eyes, mouth, etc. How does asymptomatic transmission work?
So asymptomatic transmission happens in the same way. Just a person is infected with the virus and the virus is producing particles that then travel from one patient to another, one individual to another through those droplets, except the person, because they're asymptomatic, is probably not taking the necessary precautionary measures to isolate themselves and aren't as aware of their ability to infect others. So that's how asymptomatic transmission works. In practice, it's the same thing. The only difference is the person is less aware that they are sick, and therefore, by extension, less aware of their ability to transmit the virus to other individuals. So one distinction between this virus and other recent viruses is that, for instance, in the original SARS outbreak that started in 2002, 2003, people tend to get sick very quickly upon exposure, and we're transmitting the virus usually when they got very sick. So the virus was being produced and infecting others at the stage when people would exhibit symptoms. In this case, the reason why it's spreading so quickly is that people can be asymptomatic and spread that way.
Okay, so next I wanted to ask, what is coronavirus and like what is its structure and what makes it special from and distinct from other types of viruses?
Sure. So viruses can be broken down through what's called the Baltimore classification. And this is just a simple way of designating viruses into different classes. The most basic separation is that there are first DNA viruses and then there are RNA viruses. Coronaviruses are RNA-based viruses. And so the first distinction is they're broken down into the content of their genome, DNA or RNA. Within RNA viruses, there are different subtypes. Coronaviruses happen to fall within a family that have single-stranded RNA genomes that are what's called positive sense, meaning that the genome is actually also an mRNA, that when the cell infects its host, that genome slash RNA is immediately translated to make the viral proteins. Another distinction that coronaviruses fall under is that there can be enveloped viruses, and that's what coronaviruses are. And then there are what's called naked RNA viruses. So coronaviruses fall within the enveloped single-stranded positive sense RNA viruses, and which means that they not only have a single-stranded positive sense RNA genome, but they're also contained within their genome is contained within a membrane bilayer. And so this is why soap becomes very effective in combating the virus. And this is why people are talking about, washing hands and not touching surfaces, because soap disrupts the membrane bilayer, and so does hand sanitizers. So those are two things that make this is a virus. One thing about coronaviruses that make them distinct from other enveloped RNA viruses is that they have very long genomes, about almost 10 times longer than some other RNA viruses. And a consequence of that is that they happen to not mutate as quickly as other RNA viruses. So things like the flu virus mutates very quickly. And this is why people need to get a flu shot every year. Because the coronavirus has such a large genome, something that it's evolved to take on is a strategy to when it copies its genetic material, it has now a proofreading capability to fix any mistakes or to reduce the number of mistakes that are introduced into its genome when it replicates itself. And that's just because in order to maintain a genome of 30 kilobases, it has to have some degree of replication, what's called replication fidelity. And so, so relative to other, that doesn't mean that coronavirus isn't evolving. It's just relative to other RNA viruses, coronaviruses tend to have slower rates of evolution due to mistake making during RNA copy. There are other strategies that it uses to evolve, such as what's called recombination, and that happens in things like bats, which are reservoirs or hosts to many different coronaviruses that are infecting them but don't kill them.
Thank you. So I've read that there are 2 identified strains, S and L. To your knowledge, are those all of the identified strains? And do you understand why one may be more potent and transmittable than the other?
S and L. So, I'm not familiar with the S and L designation right now. I know that there is a difference between the current virus, SARS-CoV-2, which is causing all the problems right now. Relative outbroke in 2003. That's SARS-CoV-1. In terms of those two viruses, they're closely related in some ways, but different in other ways. So their genome between the present virus and the earlier virus has about 78% match, and they bind through the same cell surface receptor, the ACE2 receptor. but the part of the virus protein that binds that receptor is different in each case. So even though they bind the same protein on the human cells to infect those cells, the way that they bind, the details are different because their proteins in that region have taken on different sequences. I know what one effort that is going on right now is to use genome sequencing, next generation sequencing, to rapidly determine the whole sequence of many different infected patient samples to see how quickly the virus is mutating as it's making its way across the globe, and try to track and see, get ahead of any of the changes that it may take on as it infects more and more people.
Thank you. As of now, does anyone that's in attendance have any questions before we move on?
Professor Ngo, so I have a question about the same difference between coronavirus and SARS back in 2003. So we now know that coronavirus is much more contagious than SARS. So what's the trait of coronavirus that make it so contagious, comparing to SARS virus?
Yeah. So they're both SARS viruses. They're both coronaviruses. Coronaviruses is a larger family of viruses, and SARS, the one that came out in 2002, is very closely related to the present one. It's just the organizations decide to name them differently. But you have a really great question as to why this virus is spreading so much more quickly, so much more contagious than the other virus. And I think there can be a couple of reasons, and I'll bring up two that people are talking about right now. One is that the difference with the original coronavirus is that people tend to be shedding the virus, meaning producing more of the virus so that it could be transmitted. The shedding happened to coincide with the same time when people were getting very sick. So in 2002, with the initial outbreak, if people were really sick, that's also when they were most contagious. And so because of that, people and medical practitioners knew that if somebody was sick, and individuals, they knew that they were sick because they were exhibiting symptoms, they were able to be quarantined and isolated from the general population. Now with SARS-CoV-2, the present virus, it can be transmitted between individuals, both before they exhibit symptoms, and people may be carriers who are non-symptomatic entirely, meaning that they don't know that they have the virus, but they're spreading it to other people. They're going into work. The droplets are, for instance, getting onto doorknobs, things like this, or they're in crowded places and they don't know that they're sick, but they're infected, they're producing the virus, or they're shedding the virus, and then other people will pick it up. And so there's a general metric that people use to describe. Let me see if I could pull up this slide, and if it's okay with you guys, I might share a screen to just show you a couple figures from that help explain this concept of what I'm trying to get at too. So there's another metric that virologists and epidemiologists look at. You guys see my screen? Yeah. One metric that people look at is called the R0 right here. And in virology terms, R0 is the, or also known as the basic reproductive number. This is a number that describes the average number of other patients someone will infect when they are sick. Okay, so somebody who has the virus that causes COVID-19, so SARS-CoV-2, and these numbers may have changed over the past week or so, but when someone is infected with this virus, they're likely to transmit it to between 2 and 2.5 other individuals. But the incubation time is long, meaning that the person can pick up the virus and the virus can be replicating in their body for a long period of time. And they may be pre-symptomatic at this stage and also able to pass it on. So the window in which somebody's actively able to pass on the virus is greater. And because not everybody who gets infected with SARS-CoV-2 will know that they are sick. So they can pass it on to other people. Whereas someone who is sick with SARS, yes, they have a larger R0 value, meaning that they are able to pass it on to a little bit, 3 people per infected individual. But the problem, the distinction is that when people are infected with SARS and when they become infectious is when they're also exhibiting symptoms. So it just happens to be the case that people tend to be infectious when they are exhibiting a lot of symptoms, and then they can isolate themselves. And the period at which a person is carrying SARS and actively producing the virus can be shorter. So there's some reasons why SARS-CoV-2 can move so quickly. One is because someone can carry it for a long time and not know that they're producing infectious particles, and then therefore not isolate themselves. So that's one parameter. Another parameter that people are thinking about as a hypothesis is that there have been some genome changes in the virus itself that have caused it to be able to more efficiently enter into cells. And this is through a genetic insertion in this type of virus that's led to a protease cleavage site in the proteins that exist on the surface of the virus. And there are hypotheses that this increases the ability of the virus to infect cells. And
Great, thank you so much. And just another question, like relating to the future. So is according to your guess, is there a possibility that there gonna emerge a virus that has all of the three high metrics, the basic reproductive number, the case fatality rate, and also like long incubation time in future? Like, is there going to emerge a super virus like that in the future?
yeah, well, first of all, I'm not a virologist. It's hard to guess how. I'd have to say that all things considered, this particular virus, it's causing a global freeze on activity right now, as you can tell, I'm sure you know very well. I think that well, a couple of things. One, we know that outbreaks will happen again, right? And two, I think that the degree of the severity of an outbreak is always difficult to predict. So if we just go back to this slide, for instance, from the one that I was showing you a few minutes ago, let's see. Ooh, well, it's gone now. But there's another related virus called MERS, M-E-R-S. Stands for Middle East Acute Respiratory Syndrome virus. And that virus has a very low transmissibility between individuals. But a difference between that virus and the 2 SARS virus is that its mortality rate is much higher, significantly higher, by 10 times. So for instance, SARS-1 and SARS-2 has a mortality rate, meaning the rate of people who get infected and then end up dying from it is for SARS-1, if I remember correctly, is about 3%. For SARS-2, guesses have been, I mean, the data is of course coming in every day, between 1 and 2 more quickly, and the incubation time is longer. Now for MERS, that was much less transmissible between human individuals, but the mortality rate was 20–30%, so much higher. So if you were getting infected with MERS, the chance of dying was much higher. It just happened to be the case that that virus happened to be harder to spread between individuals that virus actually jumped from bats to camels, and then from camels to humans. And among the camel population, it was very transmissible between individuals, camels. Why it is less transmissible between individual humans is not completely understood, but probably worth studying at this point. In terms of whether a new virus that's super dangerous will emerge, I think we're likely to run into epidemics again in the future. And what needs to happen is we need to be better prepared than we were this time. There are certain viruses that you probably remember recent outbreaks from Zika and also Ebola, and their routes of transmission are a little bit different. So for instance, Zika is transmitted through mosquitoes. So places where there are many mosquitoes, where the initial outbreak was in South America, I think Brazil, can move very quickly among a population because a mosquito can bite one individual who is infected and then by another individual to pass on the virus. Now, Ebola, there was an outbreak also recently with Ebola, and Ebola virus is incredibly dangerous, but its transmissibility is a little bit lower, and people will only transmit the virus when they are sick, and they get very sick when they're infected with Ebola. And the people who tend to pick up the virus from infected individuals usually have to come into very close contact with those people. So the Ebola virus, what we were seeing several years ago was that the most likely people to become infected with Ebola virus were hospital workers that were treating Ebola virus patients. So each virus does have its own different lethality, transmissibility, mutation rate. Being able to predict that, I guess I can't speak so much to making predictions as to what will emerge. The experts at the Needle at BU might be able to have a better sense of that. But these outbreaks have been happening since the beginning of time, and so we're likely to see one again. And it's just a matter of being prepared and listening to experts and getting tests off the ground so that we can respond quickly.
Thank you.
Thank you so much.
I have another question. What is the biological difference that allows one patient to be symptomatic and another to be asymptomatic?
That's a great question. That is a great question. My answer for that is we have no idea right now. So for instance, you might have seen some statistics showing that we know that older people are more likely to have severe or are more of an at-risk population. Now that so much isn't so much of a surprise, because we know that older people are more likely to have severe reactions to the flu and other respiratory diseases. But questions like, why is it that some older people have worse times than other older people? So for instance, older men this virus tends to be infectious, has a higher mortality among older men than it does other groups. Now, what we're learning is that people of all ages can succumb to the virus, either have a very, very severe reaction, a very severe flu, and of course, young people can die from it as well. Older people, because of the age and burden accumulation of many mutations, and et cetera, and just being old, can succumb to the virus as well. But it's not clear why some people are infected and are entirely asymptomatic, or have mild reactions, or have the severe reactions. There are some hints, like for instance, people with pre-existing conditions, pre-existing respiratory or cardiovascular conditions, of course, are more at risk. But it's not so obvious right now why some people are suffering more severe reactions. And what people are doing, they're trying to collect genetic information from patients who have been infected without and got through without any symptoms, and then also from the other extreme, and try to use that genetic information to try to see what is the, if there is a genetic basis as to why some people respond this way and others do not. So that'll be interesting to see.
Thank you. So now we're going to move into the next section, looking at what are the current developments in preventative, diagnostic, and therapeutic developments. So we can start with preventative and diagnostic. So what type of lab tests are available and such.
Yeah, so at present, right now, there are two types of laboratory, two general classes of laboratory tests that people use to diagnose the disease. One is to catch things early on, to actually test for the viral genome, the viral genetic content through PCR-like procedures. And that is important because it can see if someone's been exposed to the virus, and it can help tell us the stage of infection. Now, eventually, someone will get infected, and if they're lucky, they'll recover and not get, most people will recover, and then they'll develop immunity to the virus. And then, if someone was infected with the virus last year, any virus, and then you try to test for the genetic content, this test will probably show up negative. So when you test for viral genetic, in this case, it tells you whether the person is producing virus, essentially. And that can give an indication of the stage of virus. Now, that's true for this virus because it's what's called a non-integrating virus. Its genome does not integrate into the host genome. Now, in HIV, that genome of HIV, it actually becomes integrated into the host genome. So therefore, if you did a DNA or RNA-based detection, you could detect the virus for many years after exposure. Now, the second type of test is an antibody test. And their antibody tests will tell someone, one, if they've been exposed to it. The person might still be sick, and they can generate positive signal in the antibody test. But it also tells someone that, one, that they've been exposed, and two, it gives us a good indication whether they've built immunity to it, because the antibodies are a immune reaction that help fight away the virus from subsequent infections. And there are two types of antibodies that people test for. One's called IgM, for immunoglobulin M, and that's a type of antibody that's produced right immediately after exposure. And then the other one is, I think it's IgA or IgG, and that's the longer lasting antibody that will continue to circulate in the body to provide defense against future infections. Now, the difference between the nucleic acid tests, looking for the genome, versus the antibody tests is that nucleic acid tests can detect whether someone's has virus in their system right now. Antibodies will take a time to build up in the body. The body has to mount an immune response, and the antibodies have to be selected, generated, and secreted. Testing by nucleic acid will give you a head start in terms of identifying people who have been exposed to the virus, asking them to quarantine themselves, assessing the stage of infection, being able to distinguish whether this person just has a regular cold or whether they have this virus, prioritizing medical care, etc. And then the antibody test is going to be very important in this next step as well, because then it helps us identify who's already been exposed, whether they're how long ago they were exposed. So if they have a lot of IgM, then that means they're probably recently exposed. If they have a lot of IgA or IgG, I forget which one. If they have a lot of the other one, the longer lasting antibody, it means that it's been a longer while. And that's important because it helps us identify people who could be producing antibodies to help fight off future infections. And you might have heard in the news that people are donating their blood, and that blood is now being evaluated to see if a transfusion to an infected individual could help boost their immune system to fight against infection. We could also analyze their antibodies, the antibody sequences. And I think in Italy, where it seems to be, the curve seems to be lowering, going down in terms of rate of daily deaths, they're starting to consider identifying people who have immunity to it and allowing those people to go back to work. You know, they've been exposed. You wouldn't expect them to get sick again.
Thank you. And then just in case anyone is not familiar with PCR, could you give a quick rundown of what PCR is and how?
Sure. So PCR is a way of where if you have it's a way of making many copies of genetic material. So if you have DNA and you want to know, and you like, so for instance, if we're going to talk about like a crime scene investigation, right? Sometimes people are genetically linked to a crime scene because, you know, they left a bit of their DNA, some of their hair, something like this. And so DNA stands for polymerase chain reaction. And the amount of DNA that's on, you know, a crime scene may be very minuscule, but we can make many copies of that DNA using the polymerase chain reaction to use the same enzymes and chemical building blocks that cells use to copy their DNA when cells divide. But we do that in a test tube, and then we make many copies of DNA, and then we can determine its sequence. And in the case of viral RNA test, we take the RNA, change it into DNA, and then make many copies of it. And we can then detect whether the viral DNA was there and use based on its genetic signature that's distinct from the regular human DNA.
Thank you. Finally, what are some current developments in therapeutic targets, such as vaccines and drug development?
Yeah. So the first thing that people do when there's a viral outbreak is to test the existing drugs because we might have stocks of them around. And many of these viruses use mechanisms that viruses share in common. So for instance, RNA viruses copy their genome to produce more RNA genomes, and then so they can package them into new viruses. So there are some drugs that will work against multiple different viruses. So some of the drugs that are being tested right now for treatment of this infection are things that have been used against or developed against Ebola virus, hepatitis C virus, which are also both RNA-based viruses. And they target that step of copying the genome to produce more RNA. And that step is unique to those viruses, and humans don't copy their own RNAs in the same way because we have DNA-based genomes. So those drugs are convenient in that they can be applied to many viruses, and that the people don't tend to make people too sick because they are selective for viral proteins, viral machinery, and not for human proteins. There are other drugs that are selective for human proteins. So for instance, the infection process of this SARS virus and SARS-CoV-2 requires certain proteases from the host to cleave the virus so that it can enter into the cell. And if you inhibit those proteases, the human protease with drugs, you can lower the efficiency of infection. And then there are other strategies as well. You may have heard of chloroquine, which has been used against hydroxychloroquine, which has been used against to fight malaria, and that works in different pathways. It prevents. People don't know exactly how it works, but what it does is it interferes with the trafficking of vesicles inside cells, which the virus relies on to mediate infection and produce new viral particles. So that's why some people think that a anti-malarial can also be applied towards virus. All of these things are in preliminary tests right now in the FDA and other organizations. But and the other thing that people are trying to develop now, in addition to these drugs, are vaccines, because for something like this, where it can spread very quickly and infect this many people, probably the best solution is a vaccine, because the drugs that people are producing are probably something that you want to take when you're infected, with it, so it can prevent the copying of the virus. Vaccine should, in principle, provide long-lasting immunity virus. So just that one can't get infected with that because your immune system's already been equipped to fight it.
Thank you. I really appreciate your time. And if anyone from the audience has any last questions, we'll go ahead and open that up. And I know we all have like a million. I could probably ask questions all day, but.
I just have a quick question. So it's like, it seems like rather obvious that this virus didn't just show up out of nowhere, that it must have been like incubating through animal species for quite a while before like finally reaching the human population.
Mm-hmm.
So my first question is, do they have any idea of how long this could have been around, or if it was just like, say, direct mutation from the original SARS virus?
Yeah.
And if so, do you think it's possible that like another pandemic level virus or something is currently incubating itself within some animal species somewhere just for one day to be released? Or
Yeah, we'll have to see eventually. That's soon to be figured out, but there was a couple things. One, there's this guy at University of North Carolina called Ralph Baric, B-A-R-I-C, and he's published a series of papers. He's been studying he's the leader in coronavirus biology. He's been studying the virus for decades, and he published a series of papers in 2015, 2014, and I can send them to you, where he said that the viral population that's circulating in bats was primed to jump over to humans. So this was predicted through multiple routes. It turns out the one that actually jumped was different from the one that he thought. But I think that there's definitely potential in this viral reservoir that exists in bats. Bats happen to be a good host for viruses, coronaviruses, but in terms of whether it seems like this virus did not mutate simply from regular SARS. There's actually a paper out today that predicts, that's just looking through genomic evolution, lineages and sequence information, that predicts that this virus diverged from SARS many years ago. So it's something that people think has been circulating in the bat population and then found several intermediate hosts before jumping to humans. And that's important because people don't handle bats directly so much as they do other animals. And when you have many animals clustered together, the jump from a bat that harbors many different viral strains to other organisms is very dangerous, because even though those organisms, that jump doesn't directly impact humans, the jump to pangolin, for instance, allows the virus to adapt to a new host and then diversifies it in a way that may allow it to jump into human patients because of the similarities between the protein structures. So another thing you may have seen in the news is that people have found that this current strain is not able to infect dogs, but has the potential to infect cats. These zoonotic pathogens are very agents are very dangerous in that they can infect multiple species. And I guess I don't know if that directly addresses either of your questions, but yeah.
Thank you very much.
Professor, just another question relating to the origin of the virus. Is there any valid scientific evidence on where the COVID-19 comes from? That also comes from bats, the same as the SARS virus or the the SARS-1 virus. Whether it's a contact or other wild animals.
Yeah, so absolutely. First of all, it's really hard to know for sure, but we have really good evidence. So for instance, the current SARS virus is SARS-CoV-2 has about 78% identity to SARS-CoV-1. So there's people think that those viruses have diverged many years ago. But looking at the present SARS-CoV-2 that infects humans, we can compare it to a strain of SARS that's infecting bats right now. And those two strains have about almost 95% identity between their genome sequences, or 93 or something like this. So very high identity between those two genome sequences, between something that's infecting bats that we know exists in bats right now, and we know is infecting people. In addition to that, there's a strain of SARS-CoV-2 that also infects pangolins, and pangolins were suspected to be the intermediate host. So it went from bats to pangolins, and then pangolins to people. And the genetic identity between the three virus strains that infect bats, pangolins, and people are very, very similar between the SARS-CoV-2 right now. And SARS-CoV-1 seems to be a little bit more distant from the one that's infecting those three. Now, SARS-CoV-1 is thought to have moved from bats to civet cats, and then from civet cats to humans. And those three, the viral species that infect those three also have very high similarity. But it's difficult to know for sure where the jump happened, but this type of genetic evidence gives us a good suggestion that was probably the pathway. In terms of MERS, which is a related virus, it only has about 55% genetic identity with SARS-CoV-1 and CoV-2. But in that case, it was thought to go from bats to camels, and then from camels to humans. And the reason why that was thought to be the case is that when people were studying that virus, one, they used the same type of genetic analysis, and two, when they looked at sick camels and collected their blood, the camels had antibodies that would bind the virus, indicating that the camels were likely exposed to the same virus. And also, the people, they had very good tracking of people who were getting sick with this virus because it was very severe, the mortality rate was very high, and people knew that when they were sick. And it turns out that in many cases, or in several cases, it was a person who had directly come in contact with a camel that was later found out to be infected. And so that was, that's not direct evidence, but that's a good indication that the intermediate host was were camels that were spreading this virus.
Thank you so much.
So yeah, well, if you guys could tell me a little bit about the organization, I'd be interested to know what your group does and how you guys meet and what type of functions you guys carry out.
Yeah, absolutely. So TIMI Global Health is, we're a smaller club that focuses on advocacy, fundraising, and service. So our major project each year is our medical service trip. And we have a long-term relationship with our partner community in Mao in the Dominican Republic. And TIMI Global Health itself is a national organization with university chapters around the US. So every 3 months, a different chapter will visit them, but they also have local doctors and community health workers who are in the community year-round. We actually had an advocacy meeting on coronavirus in early February, I believe. None of us really saw this coming, but we were tracking it pretty early on. And we also we hosted a conference on immigrant and refugee health this past semester in the fall. And we had a few professors speak from the engineering department, from political science and stuff like that, just so people could see the multidimensional aspects contributing to public health. Yeah. The idea behind our organization.
Yeah, that's great. What you guys do is really important, and I think it's good to think about these things from multiple points of view, both the science, the engineering, the social aspects and the political environment as well, because yeah, we've seen these things happen before. Most of the times in the news, and it's always seems like a world away. But I think this, at least in my lifetime, this is the first time where I'm living in somewhere where the active in this local region, right? And so in previous outbreaks, it was they were more isolated and confined, but this one clearly is spread across the world. And so I think coming out of this, everyone will have a responsibility to learn more about how we can prevent these types of things in the future, and slow, because it will happen again. It's just all a matter of getting ahead of the virus before it makes too much damage, or whatever it is. So.
Thank you. And thank you guys for attending. I will be posting this on our account.
Thanks for having me, and hang in there. And if you're sophomores, juniors, freshmen, when we're back on campus, feel free to swing by and say hi, and be happy to talk to you guys again.